from the immune system. Many of them synthesize a capsule. Interestingly, sometimes they don't synthesize the capsule until after the infection. So on the surface of the body, they don't have a capsule. But once they've infected the body, they start synthesizing a capsule. Of course, you can imagine that that capsule in itself is going to provide some protection. It's going to inhibit phagocytosis, may facilitate attachment to artificial surfaces, so this may be why we see the fact that it can spread so easily when attached to sutures, because it starts producing this capsule. The microbe produces a number of extracellular products. So it secretes a lot of proteins that can interfere with the host defense mechanism. Leukocytins. These are going to be enzymes that kill white blood cells. Hyaluronidase. This is an enzyme that causes the connections between cells to separate and loosen. So it allows the bacteria to move through. Proteases and lipases. These are enzymes that are going to break down proteins, break down fat. So the microbe has a number of mechanisms that it can use to help avoid host defenses. One other thing, it can bound coagulase. This is the enzyme that is necessary for clot formation. And so the microbe can cause the formation of blood clots. Causes the activation of fibrinogen, and then a blood clot forms. And what is the benefit of this? The microbe can hide within the blood clot. And so another way of protecting it from being exposed to macrophages. We talked about MRSA a little bit. This is methicillin-resistant Staphylococcus aureus. In 1945, 95% of staph strains were susceptible to penicillin. Today, more than 95% are resistant to penicillin. So, a derivative, methicillin, has been used for a number of years to treat Staph aureus. This microbe isn't inactivated by this methicillin isn't inactivated by beta-lactamase, which is an enzyme that some microbes can produce that will break down penicillin, so prevent it from carrying out its function. But what we're seeing now is that Staphylococcus aureus is becoming more and more resistant to methicillin. If you are working in the health field, if you are working in a hospital, there's a 20% chance that you are currently colonized by MRSA. Doesn't necessarily mean that it's going to make you ill, unless, of course, your immune system becomes depressed. Then there's a chance it could. We see breakouts in hospitals, child care facilities, in prisons. Right now, more people are dying from MRSA than from HIV. And we are also seeing now that it is developing a resistance to vancomycin as well. We started using vancomycin in 1997. Best way to control the microbe is cleanliness. Stay away from people that have boils and other infections. There is work on vaccine, but we don't have one yet. When I was working at the wastewater treatment plant, I infected myself with Staphylococcus aureus. I had, my skin had dried right where your ears crease here, and it was itching me, and so I was doing this one day, just pulling on my ear, and so because of the skin was cracked there, it had been compromised, and I'm working in a wastewater treatment plant, you know, there's a lot of nasty stuff there, and I must have contaminated myself. A couple days later, this rash started to move across my face, and it got all the way to here before I got into the doctor. And that's about 3 or 4 days of antibiotic treatment, and it started to recede and it got away. But it scared the crap out of me for sure. I'll tell you that. Okay. So those are the bacterial, those are the infections caused by Staphylococcus aureus that I'm going to talk about. I want to move on and talk about 2 infections that are caused by vectors. First one, Rocky Mountain spotted fever. This is a disease that's caused by rickettsia. And it's transmitted by a tick. So it requires a vector for transmission. This microbe is an obligate intracellular microbe. It was studied by Howard Ricketts for a long time. Probably spent 5 or so years studying the microbe, trying to grow it in the lab. And he couldn't grow it in the lab. Couldn't grow it in the lab. He gave up and moved down to Mexico and started working on another microbe similar to it, got infected and died. So that's why we named this one after him. But the reason he couldn't grow it in the lab is because of this. It was an obligate intracellular, and he hadn't made that connection yet. So he couldn't grow it on media, couldn't grow it in a laboratory setting. So it's transmitted by a tick, and it's manifested by a faint rash of pink spots. These spots can become raised and hemorrhagic. The tick transmits it via its saliva. So as it's biting you, it's going to transmit the microbe. But it doesn't do it right away. Actually, it feeds for about 4 to 10 hours before it decides to transfer the microbe. So if you've been out hiking in the woods or something like that, if you get home, check yourself for ticks right away and find any. You can pull those ticks off before they've had a chance to infect you, hopefully. So after about 4 to 10 hours of feeding, transmitted by So after about 4 to 10 hours of feeding, the tick will transfer the microbe into your blood, where it will be taken up by endocytosis from the cells that are like the blood vessels, the little capillaries there. So those endothelial cells are lining those capillaries. The microbe will bind to those cells and then induce endocytosis. Okay, so now it's made its way inside the cell. It breaks free of the phagosome and begins to multiply inside the cytoplasm and the nucleus of the cell. So it breaks free of the phagosome and begins to multiply inside the cell. Eventually, as the multiplication continues, the cell is going to lyse, it's going to burst, and the microbe is going to be released, and it's going to spread the infection.
Was this still in the red blood cells, did you say? Or?
No, this is actually the endothelial cells that are lining the capillaries. So this is an example of a zoonosis. And we don't have a vaccine, but we can treat it with tetracycline. So one of the ways of keeping this straight from the other stuff that we've been talking about, that this is delivered by a vector. Oh, I can't push that one up. I'm sorry? The other one that we are going to talk to, the other bacterial skin infection, is Lyme disease. Again, it's spread by a vector. And the microbe that's responsible for this Borrelia burgdorferi. We first identified this microbe in 1982, but it was probably around long before that. There are generally 3 stages of the infection. It starts with a skin rash. Usually it's a skin rash that migrates in a radial fashion away from the bite. One of the ways that you can identify Lyme disease is by that characteristic rash that develops. If it's left untreated, it can interfere with the SA node of the heart, so it can interfere with the heart rhythm, and eventually, if left, it can cause arthritis, doing damage to joints. This is a microaerophile spirochete. And the thing that is interesting about it that is different than microbes in general, it has a linear genome. Remember, all bacteria we've talked about so far have been circular with their genome. This microbe has a linear genome. Again, we don't have a vaccine for it, but it can be treated with antibiotics. Actually, this treatment is a prolonged treatment too. I guess it's not easy to get the microbe. One thing I forgot to mention about rickettsia. If you remember from our discussion about mitochondria, this is the microbe that is likely the great-great-great-grandpa of mitochondria. Okay, moving on, let's talk about some viral skin diseases. So, many different viruses can cause skin rashes. You know, we're not going to be able to identify all that could possibly do that. And a lot of times, the viruses are carried via the blood to the skin. So remember we were talking about infection has to be appropriate. You know, if it's an intestinal disease, you got to ingest it. If it's a respiratory disease, you got to inhale it. We see with skin diseases though, sometimes they are transmitted either respiratory or digestive, make their way into the bloodstream, and then from there make their way to the skin. The one that we can talk about that we're all familiar with chickenpox, varicella. If you're familiar with this, it's, you know, a progression from macules to papules to vesicles to pustules. So it's just those little red bumps that develop. And the problem with chickenpox is that the lesions are puretic, which means they're itchy. And so you scratch. And the problem with scratching is that you can break them open and then expose yourself to Staph aureus and Streptococcus pyogenes. So a lot of secondary infections develop because of chickenpox. The virus takes up residence in your nerve cells, and so once you've been infected with chickenpox, you have it for life. Even though your immune system is pretty much going to keep it in check, every once in a while, as you get older and your immune system becomes depressed, it can re-manifest itself in the form of shingles